Hello, we're glad you've joined us for this live webinar, Analysis of the Transcriptome of Carriers of Pathological Variants in presenilin 1, presenilin 2, and amyloid-beta precursor protein that cause Alzheimer's disease. I am Judy O'Rourke of LabRoots, and I'll be moderating this session. Today's educational web seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. It's brought to you by Lexagen. Lexagen is a transcriptomics and next-generation sequencing company focusing on the development of technologies for complete transcriptome sequencing. Its portfolio includes innovative molecular biology kits, software, and services for RNA-seq. To learn more, visit www.lexagen.com. Let's get started. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. You can post questions to the speaker during the presentation while they're fresh in your mind. To do so, simply type them into the Q&A box, which will open when you click the green Q&A button at the lower left of the presentation window. Questions will be answered after the presentation. To enlarge the slide window, click on the screen icon in the lower right-hand corner of the slide window. If you experience technical problems seeing or hearing the presentation, just click on the support button found at the top right of the presentation window, or report your problem by clicking on the green Q&A button at lower left. I now present today's speaker, Oscar Harari, PhD, and Assistant Professor in the Department of Psychiatry at Washington University in St. Louis. Dr. Harari's research is focused on the identification of omics factors implicated in complex neurodegenerative traits, in particular Alzheimer's disease, AD. The identification of these genomic, transcriptomic, and proteomic factors and their interplay will help us to understand the etiology of the disease and to develop new drugs and their respective companion diagnostics. Dr. Harari has employed traditional biomarkers of AD, that is cerebrospinal fluid A-beta, to identify a novel cerebrospinal fluid biomarker. The discovery of biomarkers for AD is a key to identifying individuals in the pre-symptomatic stage of the disease. Biomarkers can potentially be used clinically for diagnosis and to track the effectiveness of novel therapies, but can also be used as endophenotypes for genetic studies, leading to the identification of novel variants and genes involved in AD and novel therapeutic targets. Dr. Harari's complete bio is found on the LabRoots website. Dr. Harari will now begin his presentation.
Okay, thank you so much, Judy, for your nice presentation, and thank you, the people from Lexagen and LabRoots, for giving us the chance to share our research. As you might know, Alzheimer's disease is the most common form of dementia, and nowadays there's no available cure. It is estimated that 5.2 million patients just in the U.S., and it is projected that by 2050, there will be 15 million of affected subjects. It's the sixth leading cause of death, and it is characterized by a progressive memory loss and cognitive decline. So indeed, the pathology of AD begins, it's well accepted nowadays, that it begins within 10 to 15 years before we can notice any decline in cognition. And from the pathological point of view, it is characterized by intracellular amyloid plaques and extracellular tau tangles. So in this chart, you can see the different events that it's well accepted that happens before the decline in cognition. So at the very beginning, almost 15 years before the decline of cognition, the amyloid starts propagating to plaques, which it follows by tau tangles, and that's when there's some symptoms of cognitive decline. And once the patients get that point, the neurons are already there. So all of the treatments of our efforts are towards the identification of the initial events that lead to this disease. Indeed, when in AD, we see a neuronal loss and astrocyte proliferation. That's something else that is characteristic of from the pathological point of AD. So there are different manifestations of AD. The early onset, which is characteristic in families, has the Mendelian form of autosomal dominant AD, and it's very rare. Only less than 1% of the patients have these sorts of manifestation. It's usually the case that the symptoms, the cognitive decline, appears before the age of 65, and it tends to be clustered in families. And the research community has already identified mutations, fully penetrant mutations in the APP, PS1 and PS2 genes. The vast majority of the subjects that have AD have in the late, which we call late onset AD, and the age onset is higher than 65. In contrast to the early onset AD, which happens to show Mendelian sorts of inheritance, the late onset AD has the characteristic of a complex genetic trait where we have different genes that confer different risk that not fully penetrant. They would confer different risk of developing the AD. Indeed, it also happens in families because we know that more than 40% of the cases have one or more affected the first-degree relatives. We're interested in understanding transcriptome of AD because that would give us some insights of the molecular networks that are out there by the disease or causing the disease. Even in doing the transcriptomics of skin fibroblasts, we can see that mutation carriers of APP, PS1, and PS2 variants are clustered by the transcriptome measured in the skin fibroblasts apart from the first-degree relatives. So this is indicating that an overall effect of this variant, this coding variant, has an effect on the overall transcriptome. So this was measured in the skin fibroblasts. So we are interested to see what happens in the transcriptome in the cerebral cortex, which is the region of the brain which is affected by AD. So that would let us understand how the genetic loci, which were identified by Genomewide association studies or next generation studies, and organized to common or distant networks, and then learn more about the disease and be able to generate some novel targets for drug treatments. So one of the problems while doing transcriptomics analysis is that, as I mentioned, in AD, there's a neuronal loss and astrocyte proliferation. So this different cell composition in the brain will confound any effect that we find in a transcriptomic profile. So, for example, in this chart that you can see in the top, the corner top right in your screen, you would see that here along the x-axis we have the fold change in microglia compared in the y-axis the fold change in astrocytes, and this is in mice that were induced with the inflammatory response. So you might see that in blue are the genes, the transcripts that have a high fold difference, higher than 4, in both cell types. But in green, you would see that those are the genes, the transcripts that have a differential expression specific to astrocytes. There's no difference for microglia. And conversely, you would have that the red ones are specific for microglia, no effect for the astrocytes. So all of these specific effects will be lost or will be confounding if we don't correct for the cell composition that we observe in brain. So there are different options to do that. We can do cell sorting where we isolate the specific cells from the tissue and then we sequence that, or we just ascertain the transcriptome, or we just can try to ascertain what nowadays we call bulk RNA-seq, just to ascertain the entire admixture, homogeneous tissue, and from there use some computational approaches to infer the expression of each of the cells, cell types that confer that tissue. So basically the idea is that we have a reference, we capture the signature of the different cell types by using sorting cells, and then sequencing those. So we capture the expression signature of astrocytes, several astrocytes, microglia, and neurons, and oligodendrocytes, and then with that we construct the reference panel. So with that reference panel, if we know how to employ this reference panel, we can get some additional RNA-seq from bulk RNA-seq, and using computational methods which are called digital deconvolutions, we can infer the distinct proportions that we have in the different tissues, and we can plug those different distributions into our analysis to correct for the different conformation, and then highlight and then obtain the true transcriptomic profiles that differ within non-demented control subjects and affected subjects. So the key while doing, there are plenty of different deconvolution methods, algorithms, and are publicly available. And all of them rely on an external reference panel, as I was mentioning, this expression signature of the different cell types that conform the tissue of interest. So it's kind of critical to conform a good reference panels. To do so, in the brain, there are not a lot of resources available, so we access a broad amount of publicly available cell type specific expression data. For example, we access the gene expression omnibus, and from there we obtain for mice, from the cerebral cortex of mice, 2 samples for astrocytes, 2 samples for neurons, 2 samples for oligodendrocytes, and 2 for microglia. Those are the cell types that we're interested in. Further, we were able to access in another gene expression omnibus study. We were able to access 4 more additional astrocytes, also for mice, and we also were able to access some human tissue that included fetal cortex, temporal lobe, and hippocampus. Then we realized that we could also add into this analysis iPSC, the derived cells. For example, we included from the AMPAD, Advanced Medicine Partnership AD, there's Broad Institute deposited in their portal, 2 additional astrocytes, those are iPSC derived astrocytes, as well as neural progenitor cell lines and iPSC derived neurons that were ascertained at 57 days and 100 days old. So we have a very heterogeneous collection of samples that we needed the RNA-seq data was obtained using different library preparations and different sequencing options, such as finding the depth, parents, and so on and so forth. So we decided to re-quantify everything using a same platform, and we chose Salmon, which is a pseudoliner, which is really, really fast and produce very good results. So we re-quantify the transcriptome against the AG19 assembly. So here in this plot, this is a PCA. This is reducing, this is capturing most of the variance that exists in the overall transcriptome that we are certain of our samples. So the first PC captures most of the variance, and the second PC is orthogonal to the first PC. That's why we say that the PCA analysis gets rid of the correlation among the genes of the transcript. But the important thing is that 29% of the variance that we capture in the samples would be correlated with the species of the donor of the sample. So basically, on the right, in blue color, you will see that those samples correspond to mice. On the left, in red, those samples correspond to humans. So basically, if you use these data as it is to discover markers or specific transcript that would let us pinpoint different cell types, this analysis will be completely confounded by the species of origin of the donors of the samples. So instead, what we have done, we accessed to the literature and picked transcripts or genes that are known to be specific or tagging different cell types. So we chose at least five different genes for each of the cells. So we want to confirm a very robust panel of gene or transcript that would tag the different cell types. So for example, we picked GFAP and XOXO-X9 for astrocytes. For neurons, we picked CCK and GAD1 and so on and so forth. So we repeat this principal component analysis, but just using these 25 genes, and the results are completely different. So here you would see that now we have circles for the circles correspond. I don't know if you were able to appreciate it, but the circles correspond to mice and triangles to humans, and you would see that now all of those are mixed, and the color corresponds to the different cell types. So on the extreme far top right corner, you would see that those three correspond to neurons. On the opposite side, you would see in red all of the astrocytes, oligodendrocytes are split by PC2, and microglia are split by PC3. So basically, what we can see here is that once we only focus on the genes that are known to be tagging the different cell types, we can cluster the different cell types themselves. And on top of that, what we have done is we projected RNA-seq, the transcriptome, again ascertained using Salmon against the AG19, and projected into the PCs that we learned from the cell type specific, and we saw that those were in the middle. So basically the RNA-seq, bulk RNA-seq from homogeneous tissue from brain was reflected to be conformed with these different cell types. That was the result that we were expecting, actually. So once we observed that, we said that we felt that we were ready to use any deconvolution approaches to evaluate the cell compositions in the samples of interest. So we have some other issues that we wanted to have the panel. We wanted to use machine learning approaches to pick which not only the genes but also the samples that would confer the best accuracy for the convolution. And you have to remember that I mentioned that we have different labs that produce the samples. We have different species donors of the sample. Indeed, some of the samples are from human fetals. There's some others that are coming from after surgery, the recession, and those are affected by some other diseases. So we just wanted to get rid of all of those confounding factors and get the broadest but most cohesive panel that would produce the best accuracy for the digital deconvolution. So firstly, we needed to define a metric how to analyze the performance of the different deconvolution method. So we chose to employ the root mean square error. Here you can see the formula. If for those of you that are familiar, this looks like the Euclidean distance corrected for the number of samples. So we wanted to measure, to give a systematic way of measuring the error of the algorithms, including one or dropping one of each of the samples. And then we wanted to do the same thing for each of the samples, and then with control mixtures. So basically, it's not the same thing to predict the purity of a sample, given that we know it's 100% microglia or neurons or astrocyte, compared to a sample that would mimic the distribution that we observe in brain. So we wanted to do everything. So what we have done is to follow a procedure called leave one out, this cross-validation procedure, where basically we train the models using all but one of the samples. And we train the model. That sample that was kept apart is used to test the model. So basically, we run that sample what wasn't included in the testing to see how well it is predicted. And that procedure is done for each and every sample that conforms the panel. And this is the results that you can see here that we obtained. So the y-axis explains the purity, how the percentage of the cell types that was predicted, and the colors are the different cell types that were the predictions of the different cell types. And on the left, you would see all of the astrocytes which are followed by the microglia neurons and the oligodendrocytes. So this chart is only showing the samples that we kept. Well, the root mean square was lower than 0.2. It's quite a relaxed value. So, for example, the first column corresponds to an astrocyte that was predicted above 95% of our deconvolution approach to be an astrocyte. Indeed, as you can see here in this chart, we try different deconvolution approaches. All of these are implemented in the CellMix package, which is accessible through the R programming language. And the first column, it's showing us the overall for each and every single cell type, the root mean square error for all of them. And then we also calculated the root mean square error for each of the cell types. So the implementation of CellMix of the method called semi-supervised non-negative matrix factorization produced the best, the most accurate result. The overall error was lower than 0.07, and outbeat the mean profile, which is an implementation of the PSEA algorithm, and also the digital sorting algorithm, which here we are showing it's the one that produces worse performance. So indeed, the error that we observe for neurons and astrocytes are more or less the same, which are followed by oligodendrocytes, and microglia are the cell types that produce the most accurate results. And we believe this because of the completely different evolution of microglia, that they're coming from different progenitor cells, and that's reflecting a completely different transcriptomic profile. So, as I said, these samples were coming from completely different labs, completely different library preparations and sequencing depth. So we asked what was the minimum depth, sequencing depth, the minimum number of reads that we needed to include to conform the reference panel. So what we have done is we downsample, which is basically each of the samples, the sequences from each of the samples were downsampled to only obtain 400,000 reads per sample. You have to consider that this is about like 0.5% of the samples. These samples have at least 20 million reads. So what we have done is we randomly sample 400,000 reads, re-quantify the expression, and redone all of this exercise of the leave one out procedure, re-quantify the convoluted, the cell composition, and then calculated the correlation within the full coverage and the 400,000 downsample representation of those samples. And we observed overall the correlation between those two is about 0.98, which is quite very, very acceptable. And you can see here the different cell types are color coded, and the sources are shown by different figures, by different shapes, sorry. And you might see that there's no bias, there's no specific effects. So, as I said, we were also interested in ascertaining the chimeric control or known mixtures. So basically, we wanted to know how well will these deconvolution platforms will perform when we are mimicking, we are providing a library that mimics what we expect to see in reality. So what we have done is that produce an augmented and extended Levonar procedure. So we constructed chimeric libraries, which does some reads that correspond to the different 4 cell types of interest. We re-quantify that chimeric library, deconvoluted using the remaining samples, and tested the error, again using the root mean square error. And we have performed this process many, many times just to get the distribution of the error. So just graphically to understand how is the chimeric library constructed. So for example, here you have an oligodendrocytes. Here the oligodendrocytes, which are sequenced. Those are FACS immunospan capture and sequence. And we have a bound file. This is a bound file that has each and every of the reads for that one. So we pick a number of those reads to conform a control percentage of the overall distribution of the chimeric sample. And we have done that several times with different distributions. So this is, I'm sorry, perhaps you won't be able to read this table, but this is like ideal 32 configurations that we thought that we might, using some other experiments, we thought that might make sense to find in the cerebellum and in the cerebral cortex. So we repeated that experiment, as I said, of confirming these chimeric libraries using 32 different configuration distributions of astrocytes, dendrocytes, neurons, and microglia, and did that 1,000 times just to get a good sample to avoid any error in the sampling. So in total, we did 32,000 evaluations, and because those were controlled, we knew exactly what to expect for each of the chimeric libraries. We could compare the convolution percentages against the expected ones and get the error. So again, we measured the distance or the accuracy using the root mean square error. So the overall error is about 0.07, which is decomposing specific for each of the cell types. Indeed, what we observed is that the astrocytes, the error for astrocytes is the highest one with 0.10, followed by the oligodendrocytes, which is 0.09. Again, the microglia is the one that is most accurate. We believe this because it's completely different origin of the cell type. So with those results, we are confident to use the deconvolution approach, or the convolution strategy, to test this in real data. So we access the Advanced Medicine Partnership for AD data. There are 2 studies. At the time we started this, there were 2 studies. The Mayo Clinic, which includes 270 subjects that were ascertained, the frontal cortex and the cerebellum, they were ascertained the transcriptome using RNA-seq, and those include 80 AD subjects, PSP, pathological aging, and controls. And we also access the Mount Sinai Brain Bank samples, which includes 320, which are only AD and controls, and those were measuring 4 different regions of the cerebral cortex. So those include anterior prefrontal cortex, the superior temporal gyrus, the hippocampal gyrus, and the pars opercularis. So in the discovery, what we have done with Mayo Clinic, we downloaded each of the BAMP files, the output from the sequencer. We re-quantify the expression using Solomon again, and then run from the transcription profiles, we deconvolute it to infer the cell composition for each of the samples. And then we use linear regression models to test whether there was any difference between the compositions of controls and AD. As you can see in this chart in blue, the neurons and in astrocytes the astrocytes are red colored. You can see that there's a significant decrease in the percentage of neural, indicating there was a neuronal loss. The effect is really, really high. It's like the slope of the linear regression is -13, and the p-value is highly significant. And again, there's astrocyte proliferation, which is shown here very significantly too. Of course, we learned which were the covides to correct for in this analysis, and we corrected for the gender, the age at death, and the RIN, which stands for RNA integrity number, which is a measurement of the quality of the RNA that might be affected along the laboratory preparation of the autopsy too. And the model, we just basically predicted the model is we predicted the number of the percentage of the neurons based on the disease and the covariates. So one of the great things about the Mayo Clinic is that they also produce data for the cerebellum, and cerebellum is a region, a brain region, which is known not to be affected by AD. So basically, we were not expecting to see any difference in the cerebellum. And when we have done the linear regression analysis, we observed there were no significant differences. And you can even see here that both AD and controls look more or less the same in terms of the percentages of astrocytes neurons. So once we observed this, we went for replications and quantified the AMP AD Mount Sinai Brain Bank samples. Those are more than 1,000 samples that are measuring this for different regions. And we only analyzed samples which were cognitively normal or definite AD. We left apart the probable or possible AD, which is a serial classification, pathological classification of brains in terms of AD. So we access the BAM files, quantify them again using Solomon, run our digital convolution platform, and inferred subtype distributions. As you can see in these charts and on the table underneath, for each of the 4 regions, we observe a systematic neuronal loss and astrocyte proliferations in AD compared to cognitively normal subjects. Indeed, the region where we observed the highest effect, most significant effect, is the peripocampal region for both astrocytes and neurons. So far, we were showing results using ideal sequencing environments. So we questioned whether we could also use non-optimal RNA-seq to test not only the reference panel, but also the samples that we of interested. So we wanted to characterize also the frontotemporal dementia affected brains. We have in the lab access to 25 brains. And we wanted to also analyze the brain composition of different manifestations of AD. As you might remember, I started my talk talking about early onset AD. So we have one of the largest collection of brains from people that have early onset AD. In this case, we have 25. And we also wanted to compare the late onset AD, which in this case we sequence in suboptimal conditions, 85 subjects. So we wanted to also to analyze whether there's any gene-specific effect, because our cohort is conformed for example, we have 24 subjects. We have sporadic IDs. The sporadic IDs, we call them, because there are not known variant associated that confer risks for those subjects. So those subjects are very interesting just to know what's going on there. We also have 25 subjects which have autosomal dominant AD, including 3 carriers of APP mutations, 21 for PS1 and 1 for PS2. We also have subjects carriers of bona fide mutations in PLD3, 36 of them, 22 of TREM2, which is a very interesting gene because this gene is associated, the variants that lie in coding variants in this gene are associated with AD, but this gene is not expressed in neurons but in microglia. So that's somehow showing the effect of the immune system on the disease. And we also have additional samples with coding variants in conditional genes. So we wanted to know what were the cell type, the cell composition of the different samples. So we produce suboptimal RNA-seq, aligned it using again Salmon, and deconvoluted using our platform, and observed that again there's a significant difference. I didn't mention, but all of these brains were ascertained from the parietal lobe. So again, we see a significant neuronal loss and astrocyte proliferations in the parietal lobe when we compare AD versus control. So if we put together the early and late onset AD, that's a age subset that's significantly different for the 20 controls that we have for both neurons and astrocytes. When we compare the FTDs, we see there's no difference from the temporal dimension, at least in the parietal, there's no difference in the distribution of neurons, but there's a significant difference in the astrocytes. So there's an astrocyte proliferation for FTDs in the parietal. Indeed, we can see that the clinical dementia rating is a clinical score that it's produced clinically by evaluating the cognition of the subjects, what they are like. So we tested the latest reported CDR score with the percentages and plotted those percentages. So the CDR goes from 0, which is cognitively normal, to 3, which is the worst state of the disease. So you can see as the disease of the cognitive decline progresses, so as the cognitive decline progresses, there's a decline in the percentages or the proportion of neurons, and there's an increase in the proportion of astrocytes that was significantly associated with CDR, again, with the p-value 0.04, but still significant. As I mentioned, we were interested in analyzing different manifestations of AD. We had 83 subjects with late onset AD, and we replicated the findings that we observed in the parietal of these subjects. We replicated the findings that we observed in the different cerebral cortex areas that we analyzed from Mayo Clinic and the Mount Sinai Brain Bank. But this in the reduced set, we also saw this significantly different cell type composition in AD versus controls. And we also were able to see this significant different composition in early onset AD. This only 25 subjects that significantly different. There's a lower number of neurons and proportion of neurons and increased number of astrocytes compared to controls. What really caught our attention is that within different manifestations of AD, when we compare late onset AD, the middle against early onset AD here in the left, we see that the proportion, the distribution of cell types is completely different. Although they are younger, these subjects have less proportion of neurons, that just showing that the effect in this cohort is completely different. Indeed, as I mentioned, we wanted to see if there was some sort of gene-specific effects. So we compare with similar. We have 34 carriers of PLD3 mutations or risk variants, and we compare the distribution of cell types of the PLD3 carriers to controls, and we observe that both the neuronal proportion of astrocytes were completely different controls. And we did the same thing using TREM2 carriers, the gene is don't express in microglia. And at the present time, we don't see any different cell type composition for this subject, which is really calling our attention. So we want to see whether there's some specific differences in microglia. As you might notice, I didn't mention anything about microglia because at the present time we are not observing any difference in the proportions of microglia for none of the different cohorts. We believe that given the different states of macroglia, perhaps we are not really modeling, or we are not using the proper references to model microglia, and that requires further analysis conforming reference panel using microglia in different states, activation states, but which is not available nowadays. Indeed, we analyze different, we stratify the FTD subjects by the mutations in the genes that they are lying on, and we observe that there are some specific effects for granulin that differs from MAPT or CNF carriers. Those are the three genes, one of the genes associated with FTDs. So we see that different genes or mutations in different genes are conferring completely cell compositions in the brain, in the parietal. So just summarizing this part of the talk, we analyze different regions of the cerebral cortex and also the cerebellum using publicly available high coverage RNA-seq, and we also generate our own suboptimal RNA-seq data from the parietal, and we observe specific different effects for astrocytes and neural composition. So we really want to analyze, all of this work is that we want to analyze the transcriptome profiles of different carriers of mutations or place variants in different genes associated with AD. And to do so, we need to correct for different set of compositions. So what we did in collaboration with the people from Lexagen, we produced RNA-seq for 17 carriers of mutations, PS1, a gene associated with autosomal dominant AD, and we employ the Lexagen Quantifying 3' messenger RNA kit and produce 10 million reads with 50 base pairs, single end. And again, this is from the parietal. And we re-quantify the expression levels using, again, salmon with specific. We didn't use a gene because this kit is capturing the 3' end of the transcript. So we model only the 3' end of the transcript as a reference for salmon. And again, we only sequence PS1 carriers, so we try to see how was the cell distributions in these subjects. So we analyze the IQR in the quantile rate. That's a measure of variations within each of the transcripts, but it's robust for outliers. So we observe that among the 1% of the transcript with highest variance, there are genes that are typical markers of neurons, and also typical markers, if we extend that to the 5%, that the genes with highest IQR, we are going to pick additional markers of neurons and astrocytes. So we know that although those are very specific brains, there is some variation there. So what we have done, apply our deconvolution method, and we were able to correct for the different distribution of compositions in the brains of these participants. And what we have done, given that we only have carriers, we apply a BAIT co-expression network. So basically, all of these subjects are carriers of PS1 mutations. So we selected all of the genes that were significantly correlated to PS1, and then fed with those genes, we run the WGCNA, weighted gene correlation network analysis method with extend. Instead of using the correlation, it's going to use another metric which is called TOM, topology overlap measurements. Well, we consider not only the correlation but also the consistent correlation among the points of genes to infer this sort of networks. So here we are showing the outcome of this analysis. So these networks then was pruned. We pruned some of the links between the genes using the Genomewide database, and remarkably, all of the nodes remain in analysis, but a huge amount of links were pruned. which produce this network. And in the center of the network, PS1, it is colored, the links leaving PS1 are colored in orange. And all of these genes, if we do a pathway analysis using, for example, David, are associated with AD. The Kyoto Encyclopedia of Genomes, HSA05010, represents a node for AD and is highly significantly associated with AD even after multiple dose correction. And indeed, one of the hypotheses that we are building in the lab is the late endosome or the endosome aspect of the AD, and also we could recover that gene ontology term with past multiple dose corrections. Remarkably, we are not only capturing this network only genes associated with early onset AD, autosomal dominant AD, as would be PS1 and APP, but we are also capturing additional genes which are critically associated with the late onset AD, which are also highlighted in this chart. So, as a conclusion, I would like to say that we have derived a reference panel to infer brain cell type composition. We provide replicable results of cell composition in different studies. We consistently observe significant decrease of percentage of neuromastocyte proliferations in the cerebral cortex, but nothing in the cerebellum. Unfortunately, we didn't see any microglia proliferation of macrophage infiltration. Basically, the genes that we are using to tag microglia would also tag macrophage, but we are not observing any infiltration. We were able to infer cell admixtures from very suboptimal RNA-seq data. Again, there's a clear trend for neuronal loss and social proliferations as the disease progresses, and we learned that Mendelian forms of disease shows a huge amount of neuronal death compared to other manifestations of AD. And we also observed that TREM2 carriers do not show the same cell type distribution as other carriers. This is a result that we need to further replicate to move forward. We also can say that correcting for cell composition is critical to obtain meaningful results in co-expression networks from RNA-seq. And as I said, the networks derived from early onset AD recover genes not only associated with the Mendelian autosomal dominant forms of AD, but also with the late onset AD. Before finishing, I would like again to thank the people from Lexagen that gave us the chance to work with their kit, and LabRoots that gave us the chance to talk about our research. And I am part of the Kuchaga lab, and we collaborate also with the Karch lab. I would also like to thank our funding agencies, which are stated there, and I am happy to take any questions that you might have. Thanks again.
Thank you, Dr. Harari, for your presentation. A quick reminder for our audience on how to submit questions. Simply type them in the Q&A box found by clicking on the green Q&A button at the lower left of the presentation window. Dr. Harari will answer as many questions as time permits. And our first question is, do you still have to correct the analysis for additional covariates?
Well, that's a good question. Yeah, definitely. We are not capturing any association of the cell type conformation or distributions that shows any association with gender or age, or that are the typical cofactors that would include in transcriptomics for AD. So yes, we still have to correct for those kind of covariates.
How does the plasticity of the expression of genes in the panel alter deconvolution distributions?
Yeah, so that's a good question. Basically, there are some concerns that given, for example, the astrocyte activation, GFAP it's a gene, a transcript which is associated with astrocyte, but the level of expression, it's increased by the activation of astrocytes. So whether we include that gene in our reference panel, that might be confounding proliferation of astrocytes with activation of astrocytes. So to do robustly this sort of analysis, you need to prove that those two things are disentangled. In our case, what we have done is we repeated this analysis, leaving the co-regulated genes apart and performing again and again and again the same analysis, and we obtain similar results, showing that this reference panel is robust to the plasticity of the expression associated with biological processes.
How could single-cell RNA sequencing improve and augment your analyses?
That's a great question. Definitely. We are working heavily to include single-cell sequencing because that would let you not only to measure the transcriptome from each cell, but also from measure each of the molecules by themselves. For with that information, we would be able to do what's called deep deconvolution, which is not only predict the composition of neurons, but just to predict the composition of very specific type of neurons. So we are working heavily to getting the data to generate those kind of models.
What are the differences between a co-expression network analysis and bait network analysis?
Yeah, the co-expression network analysis is usually the case that you have subjects, contributors, samples with different statuses. So in an unsupervised way, you infer the network, and then you pick the networks that are associated using correlation, for example, with the status or any phenotype of interest. In the bait network analysis, this is because of the flexibility of this package that I mentioned, that the GCNA allows you to just analyze the transcriptional context of one gene of interest. In our case, was PS1.
Well, thank you. I would like to once again thank Dr. Harari for his presentation. Do you have any final comments?
Thank you so much.
I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today will be addressed by the speaker by email. We would like to thank our sponsor, Lexagen, for underwriting today's educational webcast. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. This webcast can be viewed on demand through December 8th, 2017. LabRoots will alert you via email when it's available for replay, and we encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.
Thank you.